Welcome, everyone, to the quarterly Federal Interagency Committee for Indoor Air Quality meeting and webinar. I want to get started by going over quickly the agenda. For those of you who've attended CIAQ before, today's agenda is going to be reversed with the presentation going first and then having the updates. So I want to get started right now with our speaker. It is my honor to introduce Dr. Clifford Ho of Sandia National Laboratory. Dr. Ho is a fellow of the American Society of Mechanical Engineers and senior scientist at Sandia National Laboratory, where he has worked since 1993 on problems involving solar energy, energy storage, water safety and sustainability, heat and mass transfer processes in porous media, microchemical sensor systems for environmental monitoring, and airborne pathogen transport modeling. Today, he is going to speak to us concerning the evaluation of airborne pathogen transport and exposure risk of SARS-CoV-2 in the built environment. Following his presentation, there will be a chance for question and answers. And then after that, we will have our agency update. The agenda can be found in the chat room, a link to the agenda for download. So just before the presentation, I'm going to hand it to Lindsay, who will give us a few reminders and logistical issues. And then immediately following, Dr. Ho will have his presentation.
Thanks, Lorraine, and thank you everyone for joining us. First up will be the IAQ Area of Interest presentation, as Lorraine mentioned. Links and details about updates will be provided in the meeting minutes, which will be available on EPA's website within a few weeks. All right, thank you. Dr. Ho, the presentation's all yours.
Thank you, Lorraine and Leslie, and I appreciate the opportunity to speak to you today on some work we've done in the laboratories, the national laboratories, to address the global pandemic. This particular presentation is going to be on the evaluation of airborne pathogen transport and exposure risks of SARS-CoV-2 in the built environment. I wanted to acknowledge the funding sources. Some of the work I'll be presenting was from Sandia Lab's Laboratory Directed Research and Development, or LDRD program, and then also I'll be presenting a summary of the DOE Office of Science National Virtual Biotechnology Laboratory Efforts, or NVBL. Katrina Waters and Tom Kerstetter, the project leads, were originally scheduled to present this, but unfortunately had to cancel in the last minute. And so I'll be presenting and representing some of that work, and again, presenting on some relevant work for how do we mitigate, understand and mitigate airborne pathogen transport and exposure in the built environment. So again, I'll summarize some of the work from the National Virtual Biotechnology Laboratory Consortium, and then I'll drill down into some of the work that I've been directly involved in regarding the impact of airflow and face coverings on exposure risk, different indoor scenarios, associated risks and mitigation measures, and then also some analyses of school buses. So starting with the National Virtual Biotechnology Laboratory, this is a consortium of national laboratories that was set up, and in this case, to respond to the COVID-19 crisis. There was a report that was recently published in December of last year that summarizes all the activities which ranged from testing to remediation methods to what we were involved in terms of viral fate and transport. So again, Katrina Waters led one area of this program, and then Tom Kerstetter led the viral fate and transport analyses. And these are available online the link is down below there, but I think the easiest thing is just Google National Virtual Biotechnology Laboratory, COVID, and you'll find it. So this particular report or activity within the NVBL consortium looked at evaluating the impacts or characterization and potential mitigation measures for airborne COVID-19 transport. So this video here shows a sneeze by Bruiba, and the issue is that even though it was thought of early on that maybe these large droplets and direct contamination was the primary driver, it turned out that there were many people being infected while indoors despite social distancing. And this video does show that for small droplets, aerosols, in the order of a few microns or less, they can be propagated for quite long distances and stay viable in the air for a period of time. So we were focused in this National Virtual Biotechnology Lab work on understanding the viral fate and transport in respiratory droplets and aerosols. Some of the practical questions we're trying to address are our current mitigation approach is effective? How does the ventilation, heating, ventilation, and cooling, or HVAC, impact viral transport and exposure? And then ultimately, how do we reopen schools, workplaces, and businesses safely? I recall that this was done back in 2020 when the pandemic was occurring. So a lot of information has been developed and some answers have been obtained. But when we started this, there really weren't a lot of answers. You might recall also that even just the transmission nature of being an airborne disease was not agreed upon for quite a while. So the expected outcome here is that we wanted to evaluate the risk of airborne exposure and measures to reduce exposure risks of viral concentrations in the built environment. Again, I'd like to acknowledge the 8 national labs here that contributed to this work. So this was a coordinated effort providing measurements and modeling, more than 50 researchers across 8 national laboratories, studying airborne transmission and the efficacy of ventilation, filtration, wearing masks, and physical barriers across a range of built environments. So to give you a snapshot of the different activities and the breadth of activities we did, Oregon National Laboratories, Brookhaven, and Sandia looked at virus and droplet and plume dynamics. Classroom and single office environments was investigated by Oregon, Sandia, Lawrence Berkeley, and Livermore National Labs. Multi-room buildings were investigated by Oak Ridge and Pacific Northwest National Labs. Even a semi-enclosed dining environment was investigated by Los Alamos National Lab. And then at Sandia, we looked at school buses. So rather than trying to present and represent the work from all these other investigators, I just want to show you a sampling of the various journal publications that have come out. And I know this is being recorded, so perhaps you could just Google the titles and look up some of the areas you're interested in that came out of this consortium of work within the National Virtual Biotechnology Lab. And I encourage you to reach out to, again, the project leads, Katrina Waters at Pacific Northwest National Lab and Tom Kirchstetter at LBL for additional information. But what I'd like to do now is drill down into some of the work that I am familiar with, some of the work that we've done at Sandia, some of it with the National Virtual Biotechnology Lab. So starting with the impact of airflow and face coverings on exposure risk, and first looking at airflow. I have a snapshot here of the journal publication that gives more details. But in this work, we did some computational fluid dynamics modeling where we wanted to understand the impact of social distancing. And remember, this was started back in March, April, May of 2020 when we all started social distancing. And first question I asked was, why 6 feet? Why is 6 feet sufficient? And where'd that come from? And of course, it came from the fact that 6 feet or 2 meters was shown in previous studies to allow for large droplets if you're talking or coughing or sneezing, these large droplets on the order of 100 microns or more, the size of a human hair. Now that'll drop out by gravity. And so 2 meters will give you that buffer. But again, the airborne transmission of smaller droplets, aerosols, that could persist for much longer distances and for longer times, wasn't clear that that was a potential mode of transmission until much later. So when we did this work, we wanted to find out what would it look like, or what would the transmission and exposure be for coughs, sneezing, talking in different environments with airflow. So we looked at these different distances, 1 foot, 3 foot, 6 foot, 9 foot, and took a look at the time integrated concentrations relative to what's being expelled at the mouth. And so what this shows on the lower right is the plume concentration if you were to cough in a quiescent air environment with no wind. This is for aerosol, so less than about 5 microns. We can treat this as sort of a plume that behaves very similarly to the bulk flow that's projected out by the expiratory event. On the left is a plot that shows the normalized cumulative exposure, and what that is again, the time integrated concentration at any of these distances divided by the time integrated concentration right at the mouth. Okay, so a value of 1 means that's what's being expelled. And at these different distances of 1, 3, 6, and 9 feet, these are the cumulative exposures. So this is tied to the risk of transmission. And a couple key takeaways here are that distance does matter and time matters. So if you increase the distance that you are away from someone that perhaps has coughed or sneezed in your direction, at 3 feet, you're a couple orders of magnitude lower than if you were at 1 foot away from the person. And then also at 6 feet, 9 feet it lowers, but not as significantly. So social distancing does help with lowering the magnitude because of basically dispersion of that plume. And then the second thing that's important is timing. It takes a while for that plume perhaps to reach you, perhaps up to a few seconds depending on how far you are away from that aerosol plume. Now we looked at other scenarios. If you were to cough, if you were downwind of someone and they cough in your direction, if you're downwind, and I should mention all of these wind airflow scenarios are at 0.25 meters per second. Maximum indoor air velocity recommended for thermal comfort by ASHRAE. It turns out that these concentrations are actually slightly a bit higher. And it's because if you're downwind of someone, that wind is sort of confining. It's confining that plume, not allowing it to disperse as much. So you do not want to be downwind of anyone, whether it's talking or coughing. If you can feel a wind or if you know the general airflow, try not to be downwind. We looked at a simulation where someone coughed into the wind. So if you're upwind of someone, intuitively, of course, that's a better scenario. So if you know where the wind is, try to stay upwind of others. You can see here that the exposure plots are showing that beyond 3 feet, there was no exposure in this particular simulated scenario. But of course, you don't want to necessarily force others to be downwind of you. So we looked at a crosswind scenario. If you can be crosswind of others, if you're talking or in a group of people, that's probably the best scenario. You minimize exposure for everybody involved. And as you can see in this plot on the left, for a crosswind scenario, anything, well, for the 3 feet and beyond, there was no exposure in these particular cases. Okay, so the takeaway here is in a group environment, if there is an airflow, you can feel it or sense it. Of course, try not to be downwind, try to stay upwind or crosswind. With regard to the impact of face coverings, we investigated masks, and this just shows a comparison with and without a mask, showing the flow trajectories of a cough, how the masks, as you might expect, will buffer that expulsion. And if you look at the concentration of water vapor coming out, this video shows that basically wearing a mask will blunt the momentum and forward propagation of very fine aerosols. Now, of course, we know that masks will filter large particles and large respiratory droplets, and that's a good thing. You know, anything above, you know, a few tens of microns can get filtered, but those smaller aerosols can basically go through most masks that aren't, say, N95 or KN95. This simulated here is it's like a filter. It's something you might use in air conditioning. A MERV 11 filter might be something close to something between a surgical and a cotton mask. What's interesting is you can see that the mask does block the forward propagation, and that could be good. You know, if you're social distancing, that definitely helps. What's interesting though is if you look at the plume, you know, that comes out, it will rise, and that may have implications for something like stadium seating, you know, or in a theater, but definitely blocks the forward momentum. And if you look again at an exposure plot here, as a function of time, without a mask, this is at 1 feet. If you're to wear a mask, the amount of exposure reduces by about an order of magnitude. And beyond 1 foot, there was no exposures observed at 3 feet or beyond with a face mask. So these notes again are saying that, you know, masks are great at filtering large droplets, but they may not be as effective with smaller aerosols, less than about 5 microns. This was published by Leong et al. Nature Medicine. But even without filtering, you know, our simulations are showing that masks can help to reduce the forward propagation and potential exposure to aerosols that get through the mask. We also did some simulations of a face shield. This shows simulation with and without a face shield. And the face shield is impermeable, so nothing gets by, but it also doesn't filter. So, you know, you can see here that some of the plume, the aerosol plume, can stick around. If they're moving, it can still be around and expose others. Okay, so in terms of airflow, face coverings, want to stay upwind or crosswind, masks definitely can help not only filter but reduce the forward momentum. I'd like to talk about some of the indoor scenarios and risks and potential mitigation measures for built environment. And we started with an evaluation of the COVID-19 outbreak in a restaurant in Guangzhou, China. I don't know if you remember this, but this was a very interesting study because it was early, very early in the pandemic. This restaurant in Guangzhou, China was with an individual who had been in Wuhan, had traveled to Guangzhou, and through contact tracing, they knew who that person infected, how long they were at the restaurant, where they sat by the cameras, security cameras at the restaurant. And so what this shows from this paper by Liu et al. Is that the infected individual in yellow was sitting at table A. The circles denote patrons, and the ones that are circled in red eventually got infected from that index patient. So what I wanted to do from this paper, I could create a three-dimensional model and investigate the impact of airflow. They felt that it was the very strong air conditioner that happened to be on at the time, but there was no fresh air. It was just an air conditioner that was on, but basically just recirculating the air. And so I was able to simulate this, as you see on the model, solid model on the right. The red person in red is the index patient. The yellow people are folks that got infected, and the green were patrons that did not get infected. So I was able to simulate the flow of air from this air conditioner on the right side of the room. They provided some information. I didn't have all the information like exact flow rate or directionality, but I did some sensitivity studies that I'll show you. But as you can see already, the flow from the air conditioner definitely blows air past the infected individual and towards the patrons, a lot of them that were infected. We simulated exhalation from that index patient as well as inhalation from all the individuals in the room. And what we found, again, here is the drawing showing where people sat and who got infected. And from our simulations, the resulting probability of infection depends not only on the resulting room concentrations, but a lot of parameters like the probability of infection, deposition fraction, viral load. A lot of these are very uncertain, but I was able to get them from the literature. And using a base case, best guess from the paper of all the parameters, I was able to replicate at least the trends. And you can see that the probability of infection increases with time. More longer the exposure, the more dosage you get in the lungs, and that's a critical parameter. Your dose goes up, probability of infection goes up. The table B ended up being the largest probability that was simulated in the actual case study. 3 out of the 4 individuals got infected, so 75%. In table A, 4 out of the 9 other individuals were infected, and so I see a lower probability, but still high. And then table C, E, and F are shown down here in the simulation that had much lower probability, which corresponds to the actual case study. So this gave me confidence that, okay, well, maybe my models are at least capturing some of the salient effects of what was actually observed in this Guangzhou restaurant. What I wanted to do is evaluate different ventilation scenarios, do some sensitivity studies since I didn't have the exact parameters, and also to look at potential mitigation scenarios. Okay, so this plot shows the flow lines from that air conditioner, and the top 2 here simulate what's called an open room. This was actually just part of a larger section of the restaurant. This wall down here opened up into a larger part of the restaurant. And then on the bottom 2, I simulated that as a closed room. To do that, I made this boundary on this side here a symmetry boundary condition. And the reason that could have occurred is because the rest of the restaurant had similar air conditioners along the wall, and so it might have made sort of this mirrored or symmetry boundary condition, and the flow in this room from the air conditioner could have been just recirculating. On the left side is a 45-degree flow going towards the tables. On the right side here is 90-degree airflow, horizontal flow. Again, I didn't know exactly the flow configuration. These plots on the right show the pathogen mass fraction. So for the person, the index patient exhaling and emitting the pathogen, this shows the pathogen mass fraction, where red is high, blue is low. And you can see a significant difference depending on what you assume for the boundary conditions. If in this closed room, which yielded a very well-mixed condition, the concentrations ended up being very high everywhere. In this top 2 open room condition, they were very spatially dependent. So one takeaway here is using a well-mixed model or assuming a well-mixed model, which is often done in studies of contaminant transport, may not be accurate. Okay, this shows all the different cases I looked at. There's 8 cases here. The first 4 are the open room. The last 4 were the closed room. Case 4 is sort of the baseline best guess case, which replicated the trends quite well. Again, the takeaways here are that the closed room, assuming just a completely well-mixed model, yielded very high, well, 100% probability of infection, which didn't occur. And the spatially varying concentrations in the open room yielded various results depending on different combinations of parameters. So I also wanted to look at, well, what if we added fresh air to the air conditioner supply? In this case, it was just recirculating. In the actual case, it was recirculating. But if we actually added fresh air, so that everything that goes into the room is assumed to come in at some percentage of fresh air, I looked at the original case, 10% fresh air coming in from the air conditioner, 50%, and then 100% fresh air. These are the contour plots of the mass fraction of the pathogen. You can see that as you add more fresh air, you definitely reduce the concentrations. And if you look at the plot of probability of infection versus these different cases of reducing or increasing the fresh air from 10% all the way to 100%, you actually do get a significant reduction in probability of infection. Of course, the trade-off is energy requirements. Bringing in fresh air increases your energy requirements for maintaining thermal comfort, potentially. So with that Guangzhou study, it basically established sort of the methodology and approach that we were taking for computational fluid dynamics modeling, gave us confidence. We were able to replicate some of the trends observed, and also the importance of a ventilated fresh air condition. We also looked at conference rooms, and this is also documented in a report that we performed. We used CFD modeling to characterize airflow and exposure risks in office spaces. Our goal is to determine the impact of plexiglass barriers, occupancy, and return vent location. So these are some model configurations we used for an actual conference room at Sandia. The upper left shows the empty room. Upper right shows social distancing with only 7 occupants. The lower left includes plexiglass barriers in front of the various occupants. That was something that many people considered and actually implemented. And then full occupancy, even though in all cases there's only 1 infected individual, shown in red. Boundary conditions, the top, we had something called troffer lights, which basically are return vents around the periphery of the lights. The supply came from diffusers at a specified flow rate and temperature. And with that those boundary conditions, we were able to simulate also the exposure, given that someone was exhaling, was infected, they were exhaling pathogens, simulated as water vapor at an average rate, and the body temperatures were maintained at a certain temperature. So this shows simulated temperatures. The contour plots are colored by temperature. You see, in this case, it was a heating scenario in the winter where the diffusers are providing higher temperature air. You can see also as the cut plane moves past the body, it shows there's a buoyant effect of the hot air rising from the body, which is not insignificant in many of these ventilation scenarios. If you look at the flow, these arrows are denoting the flow for this room and ventilation configuration. It's a very well-mixed condition. So in this case, it is well-mixed. You can see some recirculation, prominent recirculation patterns, and the flow basically going in a multitude of directions. So when we look at the concentration, the time varying mass fraction of pathogen for the various scenarios, on the left is the baseline. You can see for the various receptors in the room, we chose the same receptors in each of the scenarios, even if there are more occupants. You can see that the mass fraction increases with time. The source that was emitted by the infected individual is up here. That's sort of your reference case. What we're looking for is what scenarios might drastically or notably reduce the receptor mass fraction of pathogens that were inhaled by the receptors. It turns out that even with plexiglass barriers, didn't really change the concentrations. Having more people in the room, creating more body heat and buoyancy, didn't really significantly reduce it overall. There were some individual changes at different locations. And the only thing that helped was a modified central return. So this is the base on the left compared to using a modified central return. It really reduced the receptor locations everywhere by about a couple orders of magnitude. So that modified central return is shown here. It's like a fume hood. Think of it as a fume hood that we just installed in the middle of the room and replaced all the other return vents with that centralized fume hood. What that did was it enabled more of a once-through flow condition. So there was less mixing and lower pathogen concentrations that accumulated in the room. So that was one key takeaway, is establishing a through-flow or once-through flow condition in rooms would help reduce the mixing and concentrations over time. Well, we wanted to test this. Those were all simulations. And so we did do some tests. Again, this is in a report, a public report that's available if you're interested. We wanted to compare alternative ventilation configurations in conference rooms to minimize the airborne transmission of pathogens. We looked at different ventilation configurations, an original configuration that was well mixed, and then a modified that promoted that once through or through flow condition. We then compared measured versus simulated air velocities and fog purge rates to gain confidence in the models. The room we tested is shown here. This is a room that was in the facilities building, so they had control over various parameters in the building of this room, the flow rate. We could reconfigure the ventilation in the ceiling. So these are just different views of the conference room that was used. The two ventilation configurations and a plan view are shown here. This is the ceiling. The original vents were shown here. We had four-way diffusers, and then two return vents located in these gray boxes here. The modified vents basically, we taped off or blocked off the flow towards the center room. We made all these four diffusers flow out towards the periphery of the room. So from the inlet, all of the flow is coming towards the periphery, and then we covered the original intake return vents and popped open a ceiling tile and just made a central return. It turns out the ceiling, above the ceiling, it was just all a plenum. There wasn't any ducting, so if you pop a tile, the air would just flow, return through here, through that opening. So this video shows an example of the fog purge machine we did to look at how long it would take to purge water vapor essentially from the room with the original and modified ventilation. So this shows that with 25 seconds of the fog machine on, it did a pretty good job of pretty much filling the room with fog. Again, we did that for the two different ventilation configurations. Let it purge, let the ventilation purge itself, and in a semi-quantitative, qualitative fashion, we tried to identify when the room was cleared out with different observers. These are just some photographs of the original vents on top and the modified vents on the bottom at different times. It looks very similar. It's hard to tell, but in person, there was a bit of a difference that was distinguishable. We had 5 observers that were designated to record their observations of the visible clear out. And we also did simulations of this room. On the top is the original vents, on the bottom is the modified vents, showing some cut planes of the relative humidity or the concentration of vapor that would be in the room. Again, these are just simulations of what happened. So you can see that it starts out saturated and then it gradually lowers. If you look on the comparison of the contour plots of the vapor concentration, the humidity, you can see that the modified vents, the simulations that we show it to clear out a bit faster, just a bit faster. And that ended up being consistent with observations that I'll show you in just the next few slides. So staying with the simulations, these are the relative humidity at different locations, at 9 different locations. And again, the modified vents that promoted this sort of through flow, once through condition, purged a bit faster, and it was a bit more uniform than the original vents. Now we can average all these to make it a little more clear. So this plot here is showing the average of all those 9 locations. The solid line is the original vents, the dashed line is the modified vents. And we can see that the normalized average vapor concentrations averaged across those 9 locations was a bit lower with the modified vents. And the observers reported a time to clear out the room when it looked to be clear was about 12.5 minutes for the modified vents and 13 to 14 minutes for the original vents. This is subjective, but if you look at the 12.5 minutes for the modified vents, that corresponds to a concentration of about 13%, so let's say 10 to 15%. And if you look at that concentration for the original vent, the simulation showed that it would have taken about a minute or 2 longer to clear out, which is consistent with the observations. Okay, so basically, according to the simulations, we're down to about 10 to 15% concentration that may have coincided with when people thought it was more or less cleared, and they indicated that. So that was sort of subjective and qualitative. We also did do some quantitative measurements of airspeed at 9 different locations throughout the room. You can see the anemometers here attached to the various chairs throughout the room, and they're located here on this plan view, the locations of 9 different anemometers. We use those anemometers to record the wind speed for the different ventilation configurations, the original and the modified. The plot on the left here is showing the original vents between the model, the CFD, and the data, which is in hatched. And we can see that the trend, it differs in some locations, but it matches for the most part. And the average, which is the solid line, dashed line, matched very closely. And then if you look at the modified vents, similar behavior. The solid bar chart is the CFD simulation, and the hatch is the data. And then the lines are the average velocities, which more or less overlay on each other. So the takeaway here is the average simulated measured airspeeds at the locations matched well, and the simulated spatial trends matched quite well in the modified vents. So as far as findings, testing provided confidence in the models and the simulation methods. We did a fog purge test, and both the visualization and the simulation showed that the modified vents purged fog faster by about 1 to 2 minutes, about 12%. And the simulation showed that the modified vents yielded a more spatially uniform purge throughout the room relative to the original vents that yielded a well-mixed condition. Airspeed measurements, the average measured versus simulated air velocities at the 9 locations were within 6% for the original vents and less than 1% difference for the modified vents. So this basically gave us confidence again in the models and simulation methods. In terms of what's the takeaway from this, we believe room ventilation configurations can be modified to increase the purge rate and minimize airborne exposures. The example that we investigated in our testing was to orient the diffusers towards the periphery of the room and allow for a central return in the middle of the room. So it had the sort of once through from the periphery towards the middle. And if you seat individuals such that no one's downwind of anyone, you could have the circle seating and you minimize the exposure. So promoting that crosswind scenario that we saw earlier was effective. A note here that the centralized return should be sized to maximize air velocities while yielding acceptable noise levels and thermal comfort. Okay, and finally, we looked at school buses. Just a few minutes here, I'll go through the school buses. This again was some work we published recently. The objectives were to understand the most important factors that impact exposure risks if there's a sick child on the bus. What scenarios could reduce the risk of exposures, like number and location of windows to open. So we did some characterization and testing, visited the Albuquerque Public Schools Transportation Center, and we measured interior dimensions of the buses and measured airspeeds and temperatures during ventilation scenarios to validate the models. With regard to the characterization and testing, this is the interior of the bus shown here. We put an anemometer on the back of a couple seats. The heating unit is shown down here. This is below the row 9 under the left seat here. And we took measurements. We hummed the anemometer reading out the window so we didn't have to, so it was an unoccupied bus when we did the tests. And we had 2 locations, one at seat 5, one at seat 9, row 9. And we modeled the bus using our CFD modeling approach. We had students at every other seat. And for the exposure test, we also had an infected individual. I'll talk about that later. But for the validation of the characterization, comparison to the actual testing we did, these show simulations. On the top is a cut plane through the left row of seats, and then the bottom is a cut plane through the right row of seats. And you can see as the heater was turned on, the simulation showed, well, that heat will eventually flow through the rest of the school bus, heat up the school bus. It rises due to buoyancy, and over time it will spread through the rest of the bus. And so we compared that to the measured temperatures and wind speeds at the 2 locations. Plot here on the left shows the comparison of the temperatures. And what's important to compare is the blue line, that's the simulation with the blue dots, and then the orange symbols with the orange lines. We actually only had 1 anemometer at the time when we were able to go to the transfer. We quick grabbed our anemometer. And so initially the anemometer was placed on the row 5 on the right. We measured it, stopped the heating, quickly went in, took a few minutes to move it, and then continued recording in the back row. So 2 different locations, and they were matched quite well with the CFD simulation, actually. As far as velocities, unfortunately, the anemometer limit was 0.1 meters per second, and most of the data was below that, which at least was consistent with our simulated wind speeds at those 2 locations. Now, in terms of exposure modeling, the scenarios that we evaluated, we looked at a stationary bus, the heater on and off, different openings of the bus, and then also a moving bus, about 10 meters per second or 20 miles per hour, roughly, with, again, different openings and the heater on and off. You can see every other window open here just the back windows and the hatch, and here just the hatch open on top. With regard to the boundary conditions for the exposure scenarios, we assumed an infected child was sitting in the middle, exhaling pathogens, and then everyone else in the bus was a potential receptor. This shows a scenario where there is a stationary bus, the door is closed, so everything was closed, and you can see that the pathogen can quickly spread through the bus. The mass fraction of the pathogen is shown in the upper right here as a function of time. We simulated this for a total of 30 minutes, 1,800 seconds. Now, if you open the door, same scenario except you just open the bus door. It turns out that having that opening allows for the egress of flow, just like the through flow condition we simulated in the conference rooms. This was good. It actually reduced the overall concentrations. We reached a steady state, as you can see in the upper right. The mass fractions are much lower than before. So having openings while you're stationary, weather permitting, is a good thing. Now with a moving bus, this shows the case where just the hatch is open. Some people may think, well, it's cold, school's in session during the winter primarily, it's cold, don't want to open up all the windows. Maybe we'll just open up that hatch on the top in the middle. Well, if you do that, it turned out our simulation showed that it's just enough to sort of stir the pot. If there's an infected individual, it's just sort of creating this well-mixed condition without a lot of ventilation. There's not a lot of flow that actually goes in. So like trying to ram air into something that there's no outlet. And so there wasn't a lot of fresh air coming into that bus. It was just, again, stirring the pot and was increasing the concentrations continuously over time. However, if you open up the back windows and you had the hatch open, it was much better. The flow would go in through the hatch, and it had an outlet. It would go out through the back windows. And you can see in the upper right, the overall concentrations reached a steady condition that were much lower when you just had the hatch open. This simulation shows if you have every other window open and the hatch, or all the windows open, that's the best scenario. On the bottom, it's blue. It stayed very low concentrations. See the mass fraction in the plot here, very low. Basically, higher the air exchange, as you might expect, the lower the concentrations. This shows a condition, whoops, went backwards. What this shows is the flow rate. I thought this was very interesting. In a condition where you just have the front and back windows open, here's a quiz. If this is a moving bus, it's going about 20 miles per hour, where do you think the air will flow into the bus? Will it go in the front windows? Or do you think it'll go in the back windows? This is something you test at home. If you have a passenger vehicle, open up your windows, take a tissue, you can see which way the flow is going in and out. Think it's going in the front, or will it enter the bus in the back? Well, it tends that the air is going to go, when it goes around the bus, you have a low pressure recirculation zone near the front. So it's a low pressure zone on the outside. It's not going to want to go into the bus. Towards the back of the bus, when the boundary layer reattaches, it'll go into the back windows, and then it recirculates a bit and then makes its way to the front. Now this has implications. You might want to put at-risk individuals, perhaps in the back where there's fresh air coming in, and then as it moves towards the front, perhaps other less at-risk individuals. So this shows a plot of the average normalized time-integrated concentrations at 30 minutes as a function of air changes per hour for all the different scenarios we considered. And as you might expect, the more air changes per hour, the lower the time integrated concentrations, which is effectively your exposure risk. Anything around 20, so I recommend 10 to 20 air changes per hour is what we found as being good for really significantly reducing your exposure. So in conclusion, we talked about impact of airflow on masks. Stay upwind or crosswind of others if there's an airflow that you can detect. Social distancing can decrease exposure by an order of magnitude or more by diluting that aerosol plume. And masks can filter large droplets and decrease the momentum and propagation of smaller droplets and aerosols. For the indoor scenarios, we looked at the Guangzhou restaurant, showed that if you have airflow without fresh air ventilation, that can actually increase airborne concentrations and exposure risks. We saw well-mixed models may not accurately predict spatial variability in concentrations in rooms. And if you had ventilated conditions with fresh air, it could reduce the airborne concentrations and exposure risks. In conference rooms, the plexiglass barriers and increased occupancy did not significantly change the room concentrations, assuming we still had just a single infected individual. Plexiglass barriers, though, are still good at if you have droplets coming out, if you're talking or coughing or sneezing, those are definitely effective. If you're just looking at aerosols movement, it didn't have an effect in the simulations. A single central return vent or a fume hood decreased the simulated room concentrations by nearly 2 orders of magnitude. So having that once through flow condition can reduce mixing, pathogen concentrations, and exposure. And then for school buses, at least 2 sets of openings, say fore and aft, were recommended to maximize their exchange rates. A single opening, like the emergency hatch, it increased the airborne concentrations and exposure risks. So at the Albuquerque Public Schools Transportation Center, one thing that I was very happy about, they actually implemented this in their policy. So weather permitting, they're going to open up as many windows as possible. But in the winter, or adverse if it's raining, at least have something like the front and back windows open to promote that through flow condition and minimize risks of airborne exposure. So finally, I'm going to summarize everything up with a simple mnemonic: think fast. Okay, FAST stands for, F is face coverings. And we just described it can filter large droplets and reduce momentum and propagation of the smaller aerosols. A stands for airflow and ventilation with fresh air. S stands for social distancing and staying upwind. And finally, T is the time of exposure. We saw that, of course, with increased time of exposure, you get increased dosage. That can also increase your probability of infection. So think fast and be doing your part to stay safe in the ongoing pandemic. Thank you for your attention. I'd be happy to take any questions.
Thank you so much. I'm going to turn it over to Lindsay. That was so interesting. I really, really enjoyed it. And it's one of those times when I'm thinking, I'm sorry I'm the moderator because I've just trying to do all these little things in the background, and I would have just loved to watch that. So I can't wait to watch the replay. And don't be surprised if you get questions from me after I watch the replay. But I'm going to hand it over to Lindsay or Christina to ask any questions.
Sure. Thank you, Dr. Ho. We got several questions coming in. So the first is, have any studies been done on cubicle suites and offices? I see a lot of helpful information about restaurants and schools, but very little on offices. What should we do as more companies request their employees to return in person to offices?
Yeah, that's a great question. And I think there may have been studies, to be honest, I would recommend going ahead and Google just those keywords and see if there's any studies. But I think the studies that we've performed can be extended to whether it's cubicles or kiosks or landing sites. Here at Sandia, we're doing that. Many people have moved to a hybrid or telecommuting situation, but they are having kiosks, landing sites at work. And I think the same general conclusions apply. You want to, if you can, ensure your ventilation is such that you've got more of a through flow or once through condition, as opposed to the conventional well-mixed condition with lots of diffusers, lots of return vents. And the positioning of these things, if you I actually have another presentation called Ventilation Awareness, where we took photos of different restaurants and scenarios throughout Albuquerque. It shows you how to recognize what a supply vent or a diffuser looks like and what the return vents, which are typically just grills, look like. Given that you don't want to be downwind of anyone, as we saw, trying to not position yourself between the return vent and anybody else is just a general rule of thumb that you can abide by. So try to Google some specific scenarios. If you don't find some studies, try to stay with some of these general rules of thumb.
Great, thank you. We have a couple of questions about school buses. The research on school buses is very interesting. What is being done to let schools know the most effective strategies to ventilate school buses to prevent the spread of infection?
Yeah, well, I think within New Mexico, I worked with our transportation, Albuquerque Public Schools transportation director, Royce Binns, who's a co-author on that last paper on school buses. And I was just really pleased that he was so welcoming of doing the tests and the modeling and adopting the recommendations. We had also talked with some other transportation directors in the surrounding communities, the Rio Rancho community. He had mentioned something about perhaps bringing this up into the larger New Mexico board. In terms of what can be done, I'm hoping if we, as far as socializing the results of that work, in terms of the windows that can be opened, having, at least fore and aft if it's cold or raining, but just maximizing that throughful condition and the air change rate. However we can socialize it, I'm not sure how best to do that. Again, I tried to work with the transportation center directors, and they've adopted it for Albuquerque public schools, but yeah, I'd love to be able to spread the word a bit more.
Thank you. This is the next question. On school buses, I recall that those in the back of the bus may get a big dose of diesel exhaust fumes. The model seems to show this. I take it that this study did not consider diesel fumes. Should we be buying electric school buses without emissions?
Yeah, well, I think that'd be great. I mean, I think we're definitely electrifying our vehicle fleet, which includes buses. It can include buses, even larger transport vehicles like trucks can be electrified. So definitely going electric is good. And to the point about having diesel fumes being brought into the bus, yes, that's a possibility, which I did not model. But so I assume that the air outside the bus was clean, but certainly there have been studies, I've definitely seen studies, even in other countries in India, particulates, not only these emissions from the bus itself, but just pollution from the outside coming in. So that is yet another consideration. My study only looked at just the pathogen being expelled from a student inside the bus, assuming that the air outside the bus was clean.
Thank you. How should we avoid infection with an airborne virus and continue to live, quote, normally? What should we do? And is this the new normal? Yeah.
I would have thought that this would be 2 years in already. So I guess the new normal is perhaps what we're living with, at least for the time being. If you're at risk, even if you've been vaccinated and boosted, I still recommend face coverings. If you're at risk, I think many here in Albuquerque, in New Mexico, we still have face covering mandates indoors. Many states are relaxing that, and depending on your personal situation, you can make your own assessment about wearing masks. I do think that, based on the data that I've seen and the modeling that we've done, I do think masks are effective. Trying to keep in mind just simple things about ventilation and airflow. If you're outside, you can feel the air. Try not to be downwind of anyone if you're talking to a friend or others or colleagues, or even indoors. Try to stay upwind or crosswind so that you're not upwind of anyone else. These are simple things that I think aren't that much of a nuisance. So if we call that the new normal, then perhaps so be it. Perhaps there'll be a time when, Omicron fades and hopefully there's not another variant. That remains to be seen.
You mentioned masks. Did the study cover different kinds of masks? For example, are N95s more effective at preventing forward propulsion of aerosols than a cloth mask?
I did not do the studies on different masks. I know that I've seen studies on different mask types on their effectiveness. Not sure if it was on the hydrodynamics and on the propagation, but I can probably say with fair confidence that, yeah, having an N95 would be just as effective as these other types of masks, if not more effective at reducing the forward propagation and momentum as well, as doing a better job at filtering. So the whole point of N95s is getting down to less than a few, being able to filter out at the micron level and submicron level, as opposed to surgical masks or cotton masks, which are not as effective at filtering out those smaller aerosol particles. So I didn't do the studies on different types of masks. I have seen publications on different types of masks at the efficacy of reducing virus transmission. I'm not sure about the hydrodynamics, but you can, again, Google some of this information and find something. And I should mention, whereas you could Google stuff, and normally if you don't have a subscription, you might not be able to get a paper. I think there's been a lot of exceptions so that if it's pandemic related, a lot of these papers are accessible nowadays.
Thank you. For the restaurant case, how was the infection risk probability calculated?
Yeah, good question. And I have actually some backup slides here that show that. So in that paper that I referred, you can take a look. It actually has it located there. But in general, the infection, the probability of infection has been studied before. And basically, it's a function of your infectivity of the pathogen and the dose. And so in general, the higher the dose, the higher risk of probability of infection. If you take this simple exponential equation here, and you look at the actual components, R is sort of this, the probability of infection is a function of time and location is on the left. The infectivity rate depends on the pathogen. The infectivity per pathogen, something called TCID50. There's the viral load. What's the pathogen level per milliliter of respiratory fluid? The density of the mucus. Vapor concentration, this is after it's in the air, what's the concentration? The fraction of pathogens viable over time when they're in aerosol form. The inhalation rate, how much you're bringing in over time. And then also a deposition fraction in the lungs. All this was considered. Now, I just took information from the literature that I could find. A lot of the information wasn't available for SARS-CoV-2, but I used relevant information as much as I could. And I plugged that all into that model of probability of infection. And ultimately, with the concentrations from the CFD model, the computational fluid dynamics model for the different scenarios, how much concentration was in the air for all those different scenarios investigated, that was plugged into that exponential equation, that integral equation, and I was able to calculate a probability of infection. The most important variation, by the way, is this viral load. I mean, this can vary by orders of magnitude, depending on the state of the patient, how sick they are, whether it's upper or lower respiratory tract. There's a lot of variability here. That was one key takeaway. By the way, I did this in a probabilistic fashion, so I did some probabilistic modeling, varying all these parameters. You might see here, this is all uncertainty distributions. So I was able to try to identify what are the most significant factors. Viral load is certainly one of them.
Thank you. Was there any work looking at modifications of ventilation in conference rooms and occupant comfort concerns?
Yeah, so that's a good question. We only looked at the impact on reducing potential pathogen concentrations and exposure. Maintaining thermal comfort is certainly a concern, and it may require additional energy if you're doing a ventilated once-through condition, but we didn't do a techno-economic study on that trade-off.
Thank you. Getting a couple more in. The next one is, how is your modeling different than what has been known? The commenter quoted, the power of ventilation to stop an epidemic is well known, from Dr. Harmon Knox Root. Well,
I think since this the pandemic started in 2020, there's been dozens and dozens, if not hundreds of studies. And so I don't want to claim that my simulations are absolutely unique. I think the comparison, the publications, I think, in journal articles speak for their uniqueness. Evaluating the Guangzhou study to help validate, build confidence in the models. Evaluating and sort of just telling a cohesive story of mitigation measures is perhaps, I think, my contribution overall. Certainly understanding that ventilation can help with minimizing airborne exposure and risks has been known, but specifically what ways, what methods can be implemented, not only for indoors, but for school buses. I think a lot of that had not been really fully investigated. But certainly I think from the comment, absolutely, there's a lot of stuff out there in the literature, a lot of it may be similar and complementary. But I'd just say, the more the better. In this time of the pandemic, there's a lot of efforts going on simultaneously. And while there might have been overlap, we can just use that to build and draw lessons learned. All the better.
Thank you. We have a few more questions. The next one is a little bit of a long one, so bear with me. Thank you and the National Labs for this excellent public research. Many of the slides noted the relative humidity in the modeled and simulated environments. Did any research examine direct effects of relative humidity on SARS-CoV-2 directly and or on human immunological response? The interactions of ventilation and relative humidity are complex, and human immune system function is affected by relative humidity. Is there any research examining the intersection yet to consider how to optimize the whole system?
Yeah. So yes, there have been a lot of studies. I was not directly involved in the coupling. Certainly relative humidity, number one, affects the evaporation of droplets. In some of the modeling studies, I assumed it was aerosols already. It was already airborne. An interesting finding is that with high humidity environments, you have less evaporation, perhaps more likely to drop out, but there can be some trade-offs too, also with regard to deposition, as the commenter made. So I think there's sort of the holistic study of the impacts of relative humidity on droplet dynamics, fraction of lung deposition, and then infectivity. I don't know if there's been a comprehensive analysis. That's a good suggestion. But there have been definitely lots of publications on various effects of relative humidity on again, droplet dynamics, deposition, and other aspects in a compartmentalized fashion. It's a good suggestion to try to take a look at just a holistic evaluation.
And we had one more question about SARS-CoV-2. Have there been any studies of the viability of SARS-CoV-2 for transport through return and supply ducts of central air distribution systems? Most commercial building filters capture a small fraction of the particles, but does SARS-CoV-2 remain viable in recirculated air?
So I do know that as part of the National Virtual Biotechnology Lab, one of the labs did look at a study on the ability for SARS-CoV-2 viability as it goes through a centralized air handling system. They did show that it was possible. I think it was pretty low, but there have been studies on that, and then also the spreading of SARS-CoV-2 through a multi-room building. It is possible. My feeling, I've done some studies or evaluations of this. My feeling is that I call it dilution and deposition. The more you can allow for that or provide for that opportunity for particles to either be diluted with fresh air or to be deposited or filtered with filters in a central air handling system, that's just going to continue to help reduce the amount of concentration. So I think of whether you have a centralized air handling system or even these packaged terminal ventilation that you find in hotel rooms. There are advantages and things to be aware of in each. And again, I cover this in a ventilation awareness. It's sort of like a hands-on, it's a quiz. Where would you sit here? What would you do here? and I do believe that these effects will reduce the concentration: dilution, deposition, filtration.
Okay, and I think we have time for one or two more. Why was the airflow reduced from 1200 CFM to 600 CFM in the conference room? And what was the CFM rate of the return opening measured at?
Let's see, I think in the conference room, we held it at 600 CFM, or there was, what was that comment on? Let's take a look. Yeah, I'm not sure if the actual flow rate, the total flow rate in the room was 600 CFM. Maybe they were picking up on a note here that the velocities were very high. This is in feet per minute, and I was just sort of mentioning that the velocities coming out of the diffusers directly was very high, but by the time they reached the anemometers that was measured and used for comparison to simulations, it had come down to a level that matched quite well between the modeling and the testing. So I'm not sure, I apologize if there was some confusion, but the tests were run at 600 CFM flow rate.
Thank you. And the last question, when evaluating the ventilation in the conference room, did you assume the supply air was clean, or did you assume a percentage of the viral load made it through the filters?
Yeah, I assumed they were clean, again, because the opportunity for dilution, depositional filtration, and also it was just more clean, so to speak. It was easier to use as a boundary condition. I didn't investigate sensitivities where we said some fraction did make it back through again. I could do that. That was what I used in the Guangzhou restaurant when I was just recirculating it through the air conditioner. That was just pumping 100% of the stuff that came back to the air conditioner went right back through again. It can be done, but I did not do that sensitivity study on the conference room studies.
Great. I think that was the last question. Lorraine?
Thank you again so much. This was so interesting, and the animated models make it even more interesting. It's going to be interesting to see how we take all of this information and move into our new normal, and how this is going to impact how we deal with flu season and everything else, not just COVID. So I think this is a great body of work. We thank you, and I especially want to thank you because I know you have an extremely busy afternoon, and taking the time to do this for us, we really, really appreciate it.